Hi Leon, so this is the second lesson of the day. So this is about the structure of DNA. So I'm going to read a little bit out that I've put on for everybody else. So an introduction, DNA is a polymer, meaning it's made of many repeating units called monomers. So polymer is a big molecule, monomer, mono means a small molecule, so mono means one. In DNA, the monomers are called nucleotides. Each nucleotide consists of three parts, a sugar, a phosphate group, and a nitrogen base. Now, each nucleotide contains those three things. The only thing that will change is the base. So it could be a sugar, a phosphate, and a base called adenine, or it could be a sugar, a phosphate, and a base called thymine. So rather than learning these chemical names, we call these bases A, T, G, or C. These nucleotides, so those sugars, the phosphate and the base, they join together to form a long double helix structure of DNA. So DNA is a bit like a twisted ladder. And those bases are found in the middle of the ladder, so the bits that you would walk up. Now, the outside of the ladder, the bits that sort of holding up those rungs, that would be your sugar phosphate backbone. So that's the same regardless of what the base is. So the only thing that changes is the base. Now on a model, we would use different colours, but they are basically chemicals. They are in equal amounts, so they only bond with certain bases. So adenine, which is A, only bonds with thymine, which is T. And thymine would only bond with adenine. So the bases are found in equal amounts to each other. Cytosine, which is C, will only bond with guanine, which is G, and vice versa. So you need to come up with a way of trying to work out which one's bonded with which. So we'll go on to practice in a moment. But what I'd like you to do first is to answer these three questions. The first one is write a definition of a polymer and give an example. And the second one is what a monomer is and to give an example. The second question, what are the four bases called in DNA? So these are the letters. You only need to know the chemical names at A level. And number 3, what shape is DNA? So it's that twisted ladder and it starts with a D and the second word begins with a H. So pause it here, write your answer and then press play and I'll tell you the answers. So number 1, a polymer is a long molecule. You might say DNA is an example. A monomer is a single unit. And you could use nucleotides as an example. The 4 bases in DNA are A, T, G, and C. And the shape for number 3 is a double helix. So task 2, most of the class are going to draw it out, a picture of DNA. For you, you wouldn't need to do that in your exam. The only thing is you need to know what a nucleotide is. So that's a common question. So a nucleotide of DNA is the sugar, phosphate and the base. They might say, what do you call a phosphate, a nitrogen and sugar together is a nucleotide? Or they might say, what is a monomer of DNA? And you could say it's a nucleotide. So we've got a video to watch and then I've got a little bit of work. So I'll try and get the video to load. It's not that long. If it doesn't work, then that's fine. I'll just load it up. There's an advert first, I think, Leon. So I'll try and pause it so you can't hear the advert, and then we'll watch the video.
And a design DNA is structured like a ladder, which has been twisted to form a shape called a double helix. The sides of the ladder are made of long chains of alternating sugar, called deoxyribose, and phosphate, and the rungs of the ladder are made of nitrogenous bases. There are 4 possible bases, which are labelled as A, T, C, or G. Higher tier candidates must also learn the full name for each base. A for adenine, T for thymine, C for cytosine, and G for guanine. Everyone must learn that there are rules on how these base pairs pair up. A adenine always binds with T thymine, and C cytosine always binds with G guanine. This phosphate deoxyribose sugar and nitrogenous base unit is also known as a nucleotide. Nucleotides are the building blocks of DNA. Now, the bases form a genetic code, which provide the instructions on the order in which the amino acids should join to form a protein. So, a gene is a section of DNA which acts as a code for making a specific protein. The following is higher tier only. The triplet code refers to how 3 bases are needed to match to one specific amino acid. So, in the cytoplasm, when a protein is being made, as the code is being read in groups of 3, a specific amino acid is matched to that group of 3, and by the time the whole gene is read out, a long chain of amino acids is formed, which will fold eventually into a specific protein. Remember, a gene codes for a specific protein. This is a gross simplification of the process, but is all you need to know for the WJEC exam.
Right, I'm going to pause it there. I'm going to go back and explain it in a little bit more sense, maybe. So, in DNA, the complementary bases form, so A, T, G, and C. They're held together with hydrogen bonds. The complementary base pairs always bond together, so A and T. So, apples grow on trees and cars go in garages, so C and G. Or at AT and Gemma Collins is how I remembered it. So you might be given a set of code. You read the code in 3s. So you might be given 6 letters, but that would only be 2 triple codons because there's 2 lots of 3. So you might see it written as a triple codon. That is 3 bases together. So if you had the code A, T and G, that is one side of your sort of rung of your ladder, and it would bond with the other side, the opposite side. So A, T and G, the A would bond with a T, the T with a A, and the G with a C. And you could do another example. So if you had T or GTA, so the G would go with a C, the T with an A, the A with a T. I think the T with an A, I think I said that wrong. So what I'd like you to do is give it a go for this code. So the first one is AGG. So write down what code would go with AGG. So hopefully you've written down TCC. The next code is GTA. So can you write down the code that goes with GTA? So hopefully you've written CAT. And the next one, CCC. So that would go with GGG. CTA. So write down the code for CTA. That would be GAT. So all you need to know is they are read out in 3s. They're complementary, so they go with each other. If you had 6 letters, that is 2 codons. If you had 9 letters, that is 3 codons. Because a codon is read out in 3s, it's called a triple codon. So the next task are some exam style questions. I'll read them out, you can write the answers, and then I'll give you the answers afterwards. So we'll try and load this up, and then we're almost done. So the first question is a picture. I'll just describe it for you because you're probably not going to ever have to feel that picture. So question 1A, which structures in the nucleus of a human cell contains DNA? The next one is X is pointing towards a part of a structure of DNA. This is consisting of a sugar, a phosphate and a base. So what do we call that? If it's a sugar, a phosphate and a base, it begins with an N. The next letter is pointing towards several letters. So you've got A, T, G and C. So what do we call these? It begins with a B. Right, the next question, C. What scientific term describes the structure of DNA when it's made up of 2 strands twisted around each other? Begins with a D and a H. Next question, D. DNA codes for the production of proteins. A protein molecule is a long chain of amino acids. How many amino acids could be coded for by the piece of DNA shown in figure A? So in figure A, you've got 1, 2, 3, 4, 5, 6, 7, 8, 9. You have got 9 letters. So how many amino acids base pairs would you have altogether? Do you have 2, 3, 9 or 18? Sorry. That's great listening to me cough. Right, E. Give 2 benefits and understanding of the human genome. The next one, question 2. When humans reproduce, chromosomes and genes are passed on to the next generation. In each of the following questions, draw a ring around the correct answer to complete the sentence. So A gene is a small section of, is it cellulose, DNA or protein? B, the sex chromosomes in the human male are they XX, are they XY or YY? You learnt this in year 9. C, most human body cells contain, is it 23 chromosomes, 46 chromosomes or 92 chromosomes? The next question, the number of chromosomes in a human gamete, so sex cells, is it the same number as in body cells? Is it half the number as in body cells or is it twice the number as in body cells? The last one, D, gametes are produced by, is it fertilization, meiosis or mitosis? So if you want to pause the video, Leon, write the answers to the question if you haven't already, and then I'll go through the answers. So the first one, A was a chromosome, or you might have said nucleus, that's fine, or genes or alleles. The next question, I was describing a nucleotide, so the phosphate, the sugar and the base. And the next question, I was describing a base, so those letters. C, so 1C was a double helix. 1D was 3, because you had 9 in total. E, so the Human Genome Project has increased our awareness of diagnosing genetic disorders. You could have gene therapy, understanding evolution or ethnic origins, understanding ancestry, tracing migration, or along those lines. Question 2A is DNA. B is X and Y for the sperm cells. I'm not expecting you can remember that. 2C is 46 chromosomes in body cells. The next one is you have half the number of chromosomes, so 23, and the last one was meiosis. So give them a go, upload what you've done, Leon, and we'll see how we get on. And hopefully we should be back in school next Tuesday. So I'll see you then.